Great. So our next distinguished speaker is Dr. Horiba. So Margit Naomi Horiba from the FDA. She's a medical officer on the gastrointestinal GI malignancies team at the FDA. She also serves as the FDA scientific liaison for hepatobiliary and esophagogastric cancers. And prior to coming to the FDA, Dr. Horiba was an attending physician at the University of Maryland Granenbaum Cancer Center, specializing in the treatment and study of GI malignancies. She will be talking to us about adaptive models of clinical trial designs and lessons learned from the NCI-MATCH study. Thank
Hello,
you.
everybody. Can you hear me?
Yes.
Oh, good. Okay. This is not the way I like to attend conferences typically, but as most of you are probably aware, we had a government shutdown recently, and the lead time to arrange travel in government requires heroics. So thank you so much for letting me participate this way. And I'm very sorry that I can't be there in person. So I took the liberty of changing the title of my talk a little bit from Lessons Learned from NCI-MATCH to Master Protocols from a regulatory perspective, not because I didn't think the topic was a good one, but because I thought that this would provide a more comprehensive look at the different options available in terms of master protocols. And hopefully this will provide an understanding. The lessons we've learned from NCI-MATCH have been nicely consolidated into a guidance document from which most of my talk is taken. And this guidance document I'll provide you the link for at the end of my talk. So I have no disclosures. Master protocols were really developed to expedite the development of anti-neoplastics by taking advantage of efficient design. There are a lot of inefficiencies in clinical development of a drug, as I'm sure most, if not all of you are aware. But we can identify certain areas in the late stage development where the process can be streamlined. So economies of scale, if you will. And as you can imagine, this can be incredibly complex and fast moving, and it's critical that these trials are well designed and conducted with rigor. So the two flavors of master protocols are umbrella trials and basket trials. And the umbrella trials test the impact of different drugs on different mutations in a single type of cancer. So think the Battle master study in lung cancer, or I-SPY2 in breast cancer, and also the lung map trial in squamous cell carcinoma of the lung. Now, on the other side are basket trials, which test the effect of a drug on a single mutation in a variety of cancer types. So this is sort of the tissue agnostic approach. And one of the older studies, or maybe even oldest master protocol, is the imatinib basket trial that led to the approval of imatinib in I believe it was 5 different tumor types. And also the BRAF positive study, which led to BRAF approval for 2 non-melanoma indications. So master protocols are intended for later stages of development in which the recommended phase 2 dose is already established. And this type of design doesn't really cover first in human trials with expansion cohorts, which is a very common flavor of clinical trial that we see. But that being said, there are circumstances in which some degree of dose finding or optimization can be incorporated, such as pediatric patients in which the recommended phase 2 dose has already been determined in adults. Some other design elements that these protocols are capable of accommodating are standard designs and adaptive designs. So the purpose of the trial can be exploratory, or it can serve in support of marketing with the appropriate tailoring of endpoints and sample size calculations and so forth. So to look at some of the general advantages and disadvantages, I think the advantages are probably obvious. In master protocols, you can use a shared control arm, which limits the number of patients that need to be used for control to develop multiple agents. You can also take advantage of centralized data capture, so there's a little bit less of reinventing the wheel with every sequential trial or every separate trial that would need to be performed. And of course, a very big advantage is streamlining patient allocation so that there's the optimization of, or the decrease of waiting times that often come with being considered for various trials. Some of the disadvantages to these designs are mostly in terms of safety. So attribution of adverse events with multiple drugs and multiple arms with no internal control. This is a common theme, I think, that we see with single arm trials. But certainly it's difficult to assess adverse events to attribute them to the disease or the treatment or some combination of both. So that's one thing that we certainly consider. The safety profile is also a little bit difficult to characterize, as we recommend that if a master protocol is being submitted to FDA, we request that that be submitted under a separate IND, just due to the complexity and the time, the effort that it takes on our end to stay on top of these studies. So when you're looking at multiple INDs with multiple people, sometimes it's difficult to see a safety signal with a certain agent. And in addition, multiple study groups permit overinterpretation of findings sometimes, and that can result in some delays in drug development. So for example, a biomarker-defined subpopulation could be identified because of multiple comparisons as a responder population, even though that is not a valid population to be studied. Okay, so basket trials, as they're commonly termed, are a single investigational drug or combinations across multiple populations. Sub-studies within basket trials are usually designed as single-arm activity estimating trials with overall response rate as the primary endpoint. Usually a strong response signal would allow for expansion in the study, and that could potentially support a marketing approval, so this makes it very attractive. And you can see below, I don't know if you can see my arrow, but with a single drug or combination, multiple tumor types can be tested. So the example I'm showing here of basket trial is the BRAF, vemurafenib, and non-melanoma cancers that I just briefly mentioned. And you can see the complexity and the tumors represented here. Most of the patients received vemurafenib monotherapy, but in the colorectal cancer cohort, after it was determined that vemurafenib alone was not effective, an additional arm was opened to include vemurafenib plus cetuximab. And this is attractive in terms of basket trial, and it sort of highlights the efficiency here. Without having to open a separate study, the combination could be studied. Now, umbrella trials, as they're commonly known, are drug or combinations that are being evaluated in a single cancer type. So umbrella trials are advantageous in that they can employ randomized control designs, and you can compare the activity of the investigational drug with a common control arm. So we recommend that the drug that's chosen for the control arm should be the standard of care for the target population. And as you might imagine, this can change over time if newer drugs replace the standard of care. So it is, to some extent, it is a moving target, but the design of the trial is able to accommodate those changes. An example of an umbrella trial is the original version of the LUNG-MAP trial in squamous cell lung cancer. So in this study, eligible patients were assigned to sub-studies based on biomarkers or molecular profiling. And they were either assigned to a sub-study based on the particular mutational findings, or if they did not have a target or biomarker of interest, they were assigned to the non-match therapy sub-study. And even within each sub-study, patients were able to be treated with the experimental therapy and compared to standard of care. And looking at sub-study E here, you can see that this these patients had a comparator arm of erlotinib, and the experimental arm was the experimental treatment plus erlotinib. So these were probably EGFR patients, and this is a nice demonstration of how add-on design, even within one sub-study, can be incorporated into these trials. So it gets more complex. NCI-MATCH is a study of multiple investigational drugs and/or drug combinations across multiple tumor types. So it's both a basket and an umbrella trial, if you will. And the purpose of this was to determine whether patients with one or more tumor mutations or aberrancies in a genetic pathway of interest could derive clinical benefit if they were treated with the drug targeting the marker of interest. And patients underwent genetic sequencing, and if they had a mutation of interest or an actionable mutation, they received the study drug appropriate for that mutation. If they progressed, they had evaluation for additional actionable mutations and flowed again back into the pathway of being considered for another agent. If they didn't have an actionable mutation, they had to come off study. So as I'm sure you're aware, there are a lot of design considerations when it comes to these types of trials. So I'd like to kind of address them one by one. The first being the control arm. So we recommend that trials in a single disease, like umbrella trials, use a single control arm to take advantage of the efficiency that this can provide. It's somewhat tempting to make a lot of comparisons between your various sub-studies and your various arms, but in general, we recommend that the comparative analysis only be conducted between the drug and the control, which is standard for most clinical trials, although I think the temptation is greater when you have these arms running in parallel. So combinations can also be evaluated within the master protocol structure. We do usually ask that there's strong scientific rationale for the combination, including evidence of synergy. We also prefer that the recommended phase 2 dose has been identified for each individual drug, and that the safety and pharmacology have been well defined and any preliminary efficacy be described. There may be some dose finding component that can be included, as I've mentioned before, so it's not completely rigid. But the safety for the combination of drugs should be established in a minimum of 6 patients. I think the biggest stumbling block for us is really isolating the contribution of each drug in a combination to the treatment effect. And this certainly isn't unique to master protocols, but the general investigational plan should generally describe the approach that will be undertaken to demonstrate the contribution of each drug to that effect. Otherwise, you've altered your risk-benefit analysis or your risk-benefit ratio and may receive all of the risks of toxicity with no expected benefit. So the issue of biomarkers is something that I think is of interest and certainly is integral to these trials. Generally, we recommend that if a biomarker development plan is being undertaken, you bring it to FDA early on so that we can provide advice on expeditious but rigorous ways of performing that. Patient selection tests should be analytically validated. The criteria should be well defined for marker positivity before starting the trial. And I think this, again, this is just good clinical trial practice that it can never hurt to repeat it. So if an investigational diagnostic is used, that's a little bit of a different story, and level of risk determination needs to be made. That can be according to or through the Center for Devices. That can also be submitted to the IND, and it would the risk assessment takes place under the Oncology Co-Development Program. In master protocols with sub-studies with drugs targeting multiple biomarkers, you can probably imagine that there are multiple decisions that have to be made. Who goes to what, or which patient goes to or gets allocated to which sub-study. And I think that what's really critical is that these protocols contain a pre-specified plan or an algorithm for allocating these patients. There are multiple ways of doing this. For example, the Battle-1 prioritized biomarkers and treatments based on the predictive value. Sorry, I'm having a little trouble seeing my screen here. And okay, there we go. The LungMap trial prioritized their algorithm based on something called a reverse ratio of prevalence rates, such that patients who had low prevalence markers were more likely to be assigned to a lower prevalence sub-study in order to make sure that there was accrual to all arms or to all sub-studies. So some other design considerations, I should just state that master protocols have an iterative process in which arms are added, expanded, or discontinued. There's sort of continuous data coming in, and certainly there are multiple interim analysis. So it's very important to pre-specify in the protocol and statistical plan those various conditions that require making changes, such as futility or sample size re-estimation. So if a sub-study is anticipated to be the basis for a marketing application, that to some extent that changes the study conduct, and this would require a blinded independent review of the efficacy outcome, the imaging typically. As well as having an independent data monitoring committee to evaluate not only efficacy, but also safety. There are obviously statistical considerations for these types of trials. Sample size calculation is important, as it would be in any trial. Futility analysis can be conducted, which is a very useful way to limit exposure to an ineffective drug. And also, as I said before, adaptive or Bayesian statistical methods can be used. And the statistical plan should include details on how those methods will be implemented. Some of the safety considerations that bear mentioning are in the study, a systematic approach should be undertaken for reporting. So, for example, you know, there are multiple levels of reporting to IRB, to FDA, to other investigators, and it can get quite complex. There should be a mechanism for rapid and meaningful communication and implementation of protocol amendments and that needs to take place for all communication, actually. And the burden of safety, of cumulative summaries of safety is a little bit higher with these types of protocols, and it typically takes place more often than annually. And we ask that investigators use experienced medical monitors to participate in these trials. And just a few other considerations here. I'm sure you can imagine the complexity with version control of the informed consent and protocol, the requirement for frequent and efficient communication, again, safety reporting, and in the meanwhile, all of the multiple analysis and revision of assumptions, recalculation of sample size, addition of new agents, et cetera, et cetera. So here is the link to the master protocols guidance. It's actually a draft guidance document, and this is this just shows you that there are multiple topics on which FDA guidances have been written, and these guidances reflect the thinking of the FDA at the current time. There is an open period usually for comments to the FDA before the draft guidance becomes finalized, and we certainly love to have input. So, you know, this can be actually a useful resource for investigators and sponsors. So in summary, master protocols have the potential to facilitate getting effective drugs to patients more quickly, but it does require commitment to rigorous conduct of the trial, and multiple outcomes have to be considered. You should have a plan, think one step ahead all the time, if not two or more, and pre-specify everything, statistical plan, allocation algorithms, et cetera, et cetera. In other words, have a very well thought through and carefully considered protocol to follow. So thank you so much for letting me participate. And again, I'm sorry I can't be with you.
Thank you. We understand. Wonderful presentation. Certainly helps us a lot with understanding, you know, how to navigate the complexities of these new umbrella trials. I think umbrella is the right term for batch, right, Mitesh? It's not a basket, it's an umbrella. Still trying to figure out the difference.